which is focusing on extrapolation and trial design considerations for chronic hypertension. Since I didn't introduce myself earlier, I'm Lilly Mulugita. I'm the Associate Director for Policy and Research in the Division of Pediatrics and Maternal Health at the FDA. Our first speaker is Sudharshan Hariharan. He's a Master Pharmacokineticist in the Office of Clinical Pharmacology at the FDA. And his presentation is going to be on dose response and extrapolation of efficacy from older pediatric patients.
Yeah, good afternoon, everyone. I hope you can hear me well. Yeah, first of all, thank you to all the workshop co-attendees and organizers for putting this forum together to discuss this important topic. It's been a great start to the workshop, and I'm pleased to kick off the afternoon session. I am a clinical pharmacologist by training, and I'm here to talk about a project that we did recently that explored extrapolation of efficacy from older to younger pediatric patients in the area of antihypertensives. This is my disclaimer that the views expressed in this presentation are primarily mine and do not necessarily represent the official position of the FDA. I would like to start by giving a brief overview of the pediatric extrapolation framework. Pediatric drug development has the same basic standards as for adults, that there needs to be substantial evidence of effectiveness for the claimed indications, and also that the drug needs to be safe for its intended use. So basically, the benefit should outweigh the risk. However, we can rely on the concept of extrapolation to provide such evidence in pediatric patients when the following criteria are met. That is when there is similarity in the disease, there is similarity in the drug's pharmacology, and there is similarity in the response to treatment. And similarity in the context of pediatric patients, which is our target population, and the reference population, which in most cases can be adults, but can also be another pediatric subset, like the project that I'm presenting today. And the consensus on the level of similarity of the disease, the drug's pharmacology, and the response to treatment falls on a spectrum. And we'll see that, and we'll see that in the next slide. So this slide shows how extrapolation of efficacy and the data needed to bridge the gaps falls on a continuum. And this was presented at greater lengths in an earlier presentation by Dr. Burkhardt, so we won't spend much time on this, but essentially to say that this is actually a multidisciplinary approach, and we need experts from various disciplines basically to understand and compare the disease pathophysiology, the drug pharmacology, such as the ADME, PK/PD, mechanism of action, and the clinical response to treatment, such as dose response, exposure response, relationships between the reference and the target populations. And following those assessments, whatever gaps in knowledge and the degree of uncertainty we have basically dictates what type of assessments are further needed in pediatric patients. If the diseases are clearly not similar and there are larger uncertainties, then you may need adequate and well-controlled trials in pediatric patients. And as you slide on this scale towards the right, with increasing level of confidence on similarity, the evaluations will also range from, say, using a study with bridging biomarkers or adopting Bayesian methodologies to simple exposure matching when the confidence is the highest. So with that background, I would like to transition to the focus of the workshop. Pediatric hypertension is clearly an area of unmet medical need. It occurs in 2 to 5% of all pediatric patients, and possibly even higher as childhood obesity is on the rise. And the most important point here is that if it's left untreated, it can lead to long-term consequences such as end organ damage to the kidney and the heart. While this area remains a significant unmet medical need, there are also significant challenges to drug development in this space. First, as we heard in one of the earlier talks, there is significant off-label use of drugs in children that limits the availability of patients for clinical trials. Second, there are ethical constraints on randomizing children to placebo, and often parents do not consent to having their child when there is a chance of randomization to placebo. Characterization of PK or pharmacokinetics in the youngest children is sometimes a barrier as exposures are affected by rapid physiological changes and organ maturation. And needless to say, we also need formulations that are suitable for administration to young children with adequate assessment of its bioavailability and interaction with food. So due to these significant challenges in evaluating drugs for pediatric hypertension, it is not surprising to note that we have very few drugs actually approved for use in pediatric patients. We heard again in the morning that there are roughly more than 60 drugs that are approved for the treatment of hypertension in adults, but as you can see, only a handful of them are approved in pediatric patients, and that too, most of them are indicated down to 6 years only. And there are only 3 drugs that are approved for use below 6 years of age. Enalapril is one, which is an ACE inhibitor, and then we have candesartan and valsartan, which belong to the class of angiotensin receptor blockers. So clearly, there is a paucity of approved drugs for use in pediatric hypertension compared to the options that we have available in adults. So because of these issues and challenges, we explored if extrapolation approaches can be used to support the development of more pediatric-friendly formulations. With that as the focus, our project objective was to compare the treatment response between older and younger pediatric patients, above 6 years of age to below 6 years of age. Candesartan and valsartan were the drugs of choice for this extrapolation project because, as you saw from the previous slide, only 3 drugs were approved in the full pediatric age range, and 2 belonged to the same class. So hence our choice of going with these 2 drugs, because having 2 drugs from the same class would also increase the strength of evidence. And also the type of data that we needed for conducting or comparing the treatment response, like dose response and exposure response type of data were clearly available for these drugs in both the older and younger pediatric age groups. And with regard to the choice of the reference group, we used older pediatric patients above 6 years of age as the comparator because they are clearly more closer in the age continuum to the younger patients. And even if we wanted to compare across to adults, some of these drugs, they were approved in adults long way back. So there were issues with easy accessibility of data from adult trials. So the next 2 slides shows the available data we had for this project. And I also wanted to show the study designs at a high level, just to give you a flavor of the type of evaluations conducted in pediatric patients. So starting with candesartan, we had 2 studies, one in older children and one in younger children. And if you look at the design, the older children study had a placebo-controlled randomized double-blind phase for 4 weeks, whereas the younger children was not placebo-controlled. Patients were randomized to low, mid, and high doses in both studies with mg per kg dosing in younger children and body weight tiered dosing in older children. The older children study used the adult-approved tablets, whereas the younger children study used extemporaneously prepared suspension. And importantly, trough concentrations were collected in both the candesartan studies at steady state, which enabled us to also compare exposure-response relationship. And the primary efficacy variable was seated systolic blood pressure in both the studies, but diastolic blood pressure data was also collected and available. Moving on to valsartan, we had one study in older children, but we had 3 studies in younger children. You heard from Dr. Flynn's presentation earlier about one of the studies in younger children, and that was study A2307. But as clear dose response was not demonstrated in that study, the sponsor conducted 2 additional studies, K2303 and K2306, to support approval in younger children. And with regard to the design, all the studies had a randomized double-blind treatment phase, which varied in duration from 2 weeks to 6 weeks, but it was not placebo-controlled. Instead, there was a randomized placebo withdrawal phase for 2 weeks for all these studies, except the last study, K2306. Again, the dosing approach was similar to candesartan, where patients were randomized to low, mid, and high dose groups with mg/kg dosing primarily used in the younger children, except A2307, which used body weight tiered dosing, similar to that of the study in older children, A2302. Unfortunately, no PK data was collected in these clinical studies, which limited us to compare only the dose-response relationship for valsartan. And similar to candesartan, the primary efficacy variable was systolic blood pressure. This slide compares the inclusion and exclusion criteria between the studies that evaluated older and younger pediatric patients. Because we are comparing the treatment response between the 2 groups, we were also interested to understand the similarities and differences between the enrolled population. And as you can see, there are many key similarities, starting with the inclusion criteria for blood pressure, where both naive and treated patients were enrolled, and they had to have seated systolic and or diastolic blood pressure greater than or equal to 95th percentile for age, sex, and height. We know that patients with secondary hypertension due to the conditions listed in the slide were excluded from both the older and younger children studies, specifically for candesartan. We know patients were excluded based on eGFR, but the cutoff slightly varied between studies, but mostly in the range of 30 to 50 mils per minute was the exclusion criteria. And also, you can see there are some other key similarities. But some of the notable caveats include children less than 1 year of age were excluded from both the candesartan and valsartan studies in the younger children due to safety reasons. And although we expect younger children to have hypertension due to renal complications or conditions, the 50% CKD enrollment criteria was explicitly specified only in study K2306, but not in other studies. But overall, the inclusion-exclusion criteria were broadly similar, supporting comparability of the enrolled populations across the age groups. So again, just to reiterate, our objective was to compare the dose response and wherever possible exposure response relationship between the older and younger pediatric patients. We used systolic blood pressure as treatment response, as it was the primary efficacy variable in all the studies. For dose response analysis, we did simple linear regression comparing the change from baseline to end of treatment visit for systolic blood pressure versus dose in mg/kg. And all the studies I showed in the previous slides were used in these analyses. And we looked at the relationship separated by the 2 age groups and primarily compared the slope and intercept for similarity. Again, for exposure-response relationship, we did the same thing where we looked at pooled as well as separated by the two age groups. Moving on to the results, this slide shows the dose-response relationship for candesartan in older pediatric patients represented by the red profile and younger patients represented by the blue profile. The blue triangles represent the mean and 95% CI of the treatment response for the respective low, mid, and high mg per kg dose tested in younger children. The red triangles represent the mean and 95% CI of the response, but grouped as tertiles, as the dosing was not exactly mg per kg, but body weight tiered dosing. Note that for this plot, as well as the subsequent plots, even though we are showing just the summary level data, the linear regression is through all the individual data points. The shaded areas represent the 95% confidence intervals of the mean regression line for each group. And you can clearly see that there is a high degree of overlap in the relationship, both in terms of slope and intercept, suggesting that the relationship is highly similar between the two groups. This slide shows the pooled exposure-response relationship for candesartan, combining the data from older and younger pediatric patients. And you can see that there is a linear relationship in response as a function of candesartan trough concentration. And in this slide, we separate the relationship by the two age groups. Again, the older children represented by the red profile and younger children by the blue profile. And again, similar to dose-response relationship, there is a high degree of overlap and similarity in exposure-response relationship for candesartan between the two groups. And now moving on to valsartan, in this slide, you see the comparison of dose-response relationship. Again, the interpretation of the plot is similar to that of the previous plots. For valsartan, there seems to be a slightly lower response in the younger children compared to the older children. But again, the shaded areas overlap, and we also see that there is overlap in the standard errors for the slope and intercept. So largely, we can conclude that the relationship between older and younger children is similar for valsartan. So that brings us to the conclusions. Clearly, the response to treatment is similar between older and younger pediatric patients, as seen by similarity in the dose and exposure response relationship for both these drugs. And this further lays the foundation for extrapolation of efficacy from older pediatric patients, provided that the disease is similar between the two groups as well. What are the implications of this project? So taken together, these findings can influence the pediatric program for another angiotensin receptor blocker, which may be coming in via a 505(b)(2) pathway with a pediatric friendly formulation to support an indication in pediatric patients. And for such programs, what is still required? We require a PK study or assessment to determine dosing that results in exposures that are similar to that observed in the older children. And in addition, as the safety profile for ARBs is well characterized in adults, we primarily need collection of safety data in younger pediatric patients that focuses on the known adverse effect of the drug class. So essentially, a much abbreviated program. So that concludes my presentation, and I just have a few people to acknowledge. This project was primarily done by Abigail Milake, with James and Lilly and myself serving as the co-mentors and co-collaborators. And I would also like to extend my thanks to Kirtida for all the scientific discussion and input, and all the other FDA colleagues for their collaboration and support. Thank you all for listening.
Thank you so much, Sudharshan, for that great presentation. Our next speaker is Ricardo Fonseca Martins. He's the Senior Medical Manager in Pharmacovigilance at Eli Lilly, and he's going to give a presentation on safety assessment and pediatric specific considerations.
Oh, good afternoon. First of all, I would like to say that it's an honor to be here speaking about safety in pediatrics. In this presentation, I intend to address 3 questions in these 15 minutes. The first question is about the renal growth and development and the maturation timelines for different functions and structures in kidney. The second is what the clinical trial evidences that we have in valsartan and candesartan tells us about safety and what we should take in consideration regarding the mechanism of action of antihypertensive drugs and regarding the underlying disease that causes the hypertension when extrapolating data from adults. I will focus this presentation mainly in the age group below 6 years old. That's where I think that the gap is more pronounced. So regarding the development kidney, this is just a reminder that although the nephrogenesis is completed around 36 weeks of gestational age, GFR reaches 50% of adult value around 48 weeks of post-menstrual age, but it increases fast during the infancy. It reaches 90% of GFR during the first year, and it reaches complete functional maturity around 2 years of age. The tubular transport in general reaches the maturity around 1 to 2 years of postnatal age as well. But it's good to remember that organic annual secretion just reaches 50% of the adult value or the adult capacity around 8 years of age. This is what's called glomerulotubular imbalance. So why this matters? It matters because while drugs that are renally cleared, the clearance improves rapidly during infancy, although the eGFR still may be affected by underlying disease burden. But drugs that rely on tubular secretion may accumulate. Also, electrolyte transport is immature and could bring to electrolyte imbalances. Another thing that's important to point is that the neonatal GFR is maintained by the balance between the vasoconstriction promoted by angiotensin II and by the vasodilation promoted by prostaglandin. And the RAS is upregulated in infancy for renal perfusion. So blockading the RAS system could pose a unique hemodynamic and renal risk in neonates. I will not talk about, I think that this is the older presentation, but in fact, it was already showed that we have 3 clinical trials under children 6 years old, and all of those were with valsartan. I will not go through the design because it was just showed, but I will focus on the safety profile. And in a study conducted by Dr. Flynn in 2008, the key safety findings were that there are no dose-dependent increasing adverse events across the 3 dose groups, and that SAEs during the double-blind phase were infrequent and not considered drug-related. And the drug-related adverse events in double-blind phases were comparable across all those groups. But when it goes to the open label extension period, that was a 52-week, we had a higher incidence of serious adverse events of 14.8%. And two deaths not considered related to the drug were reported, although one of those deaths were due to gastroenteritis that could pose a question regarding the hydration in these patients with kidney disease. Regarding the drug-related adverse events, it was stable when compared against to the double-blind period. In the 3rd clinical trial that was conducted, the major difference, it was already said, is that we have a chronic kidney disease enriched cohort, and we had stratification by chronic kidney disease. In this trial, we had patients distributing in a low dose and a high dose. And again, adverse events comparing low dose and high dose does not show any meaningful difference. Regarding chronic kidney disease, comparing adverse events in chronic kidney disease subgroup against non-chronic kidney disease subgroup, we also don't find no meaningful difference, although when looking at the chronic kidney disease subgroup, we had a serum potassium elevation more than 20% above the baseline in CKD group. This is critical given the immature tubular potassium secretory capacity in this age group. One thing that I would like to mention, again, this is not the updated presentation, but I include the Candesertan study here as well. And in Candesertan study regarding safety, we had patients reporting acute kidney injury. We had one patient that reported an acute kidney injury, one patient, like Dr. Flynn already said, that reported death that was probably related to acute kidney event on a chronic kidney disease, and two patients that withdraw the study, one of those because of worsening of renal function. So this shows that more than just the drug action, but the underlying condition could play a role in safety for those patients. Now, comparing what we have in valsartan regarding adults, children, and children 1 to 5 years old, it's important to note that hyperkalemia, although present in all the ages, is more mild and manageable in adults and children between 6 and 17 years old, while it can be an issue in patients that have an immature tubular function in children that are below 6 years old. And also, we have long-term safe data for adults, knowing from the cardiovascular outcome trial data, but we don't have long-term safe data in children in any age group. The maximum follow-up was 52 weeks in open-label extensions. Now, I want to call the attention that what I'm going to talk now, I'm not saying that we should use one or other drug. This is a safety talk. And I will talk about the different mechanism of actions of the drugs and what risks it could pose for the pediatric patients. This is theoretical since we don't have data gathered in those patients. So the ACE inhibitors and MARAs and aldosterone antagonists are the ones that poses the highest risk for the patients according to the mechanism of action. ACE inhibitors blocks RAS, and this could remove the afferent arterial support and increase the risk of an AKI risk. Also, aldosterone antagonists may critically amplify the risk of hyperkalemia due to immature potassium secretory capacity. And also, aldosterone has developmental role in tubular maturation itself. Beta blockers and thiazide diuretics would pose a moderate risk. Beta blockers can mask hypoglycemia in infants, and it blunts the compensatory tachycardia that's the major response in neonates and infants to the low cardiac output, while thiazide diuretics can promote electrolyte imbalance amplified by immature sodium chloride cotransporter expression and limited tubular sodium reserve. Calcium channel blockers would be the one that has the lowest risk because although it causes reflex tachycardia and gingival hyperplasia is reported, it's the one that brings the lower hemodynamic and developmental conflict with the renal stage of this population. Again, let's take in consideration now the underlying disease. And we know that the majority of kids below 6 years of age has renal disease as the major cause of the hypertension. So regarding the renal parenchymal disease, the reduced nephron mass further limits tubular electrolyte reserve. And this can amplify the electrolyte imbalances beyond what the maturation curve predicts. And the hyperkalemia signal in chronic kidney disease in the study of valsartan just reinforces it's an empirical demonstration of that. Also, RAS is activated pathologically on top of developmental dependency. So RAS blockage in these patients could induce an AKI in a synergistic manner, not in an additive manner. Renovascular hypertension, although rare, is a cause that is the underlying disease for 10% of the patients in this age group. And we saw that those patients were excluded from clinical trials. And blocking the RAS system in those patients removed the only compensatory mechanism for maintaining GFR in the cyanotic kidney, causing acute renal failure. So even the safe data from the chronic kidney disease pediatric populations that we have in valsartan and candesartan and from adults could not be used here in this population. Coarctation of aorta is another common cause for those patients. And paradoxical hypertension, post-repair that occurs as a crisis of hypertension is the major issue. And we don't have a lot of data in IV antihypertensive drugs in this group. So what we know, what we need. The evidence now supports that we have short-term data in our IRBs for 1 to 5 years old patients, although we don't have any long-term data for this drug or any other class drug for antihypertensive treatments. And we don't have data in neonatal patients as well in any type of drug. The organ system being treated is still under construction in this age. So a negative short-term trial does not mean that we have a long-term developmental safety. So the messages that I want to leave here in this presentation is for the patients under 6 years of age is that kidney is not mature. We have some randomized clinical trial evidence in short-term data and safety for patients between 1 and 6 years old. We don't have any long-term evidence, and we don't have neonatal evidence. We don't have any other class with randomized clinical trial evidence. The mechanism here must guide the risk. And again, this is all about benefit-risk balance. And I'm talking about the risk. I'm bringing the risk part to the table. Of course, the benefit should be weighted here to understand which class we should use and when we should use those medications. The underlying diseases should amplify the risk, and it should be taken in consideration. And short-term trials are necessary, but we have to gather long-term safety assessment for those patients as well. Thank you.
All right, so we're going to pivot a little bit now and move into a panel discussion. I'm going to invite the moderator, Jennifer Lee, to come up. Jennifer Lee is the professor of pediatric cardiology at Duke Department of Pediatrics. We also have multiple panelists who will be joining us, some virtually and some in person. So I'll go ahead and invite Tammy, McLoughlin, Brady, and Fraz Ismat to join us for the panel discussion.
Okay. Get the slides. Okay, good afternoon, everyone. So we're going to have a panel discussion on lessons learned from conducting clinical trials in pediatric hypertension. So I'm just going to start out with the first. Most of all of you are aware of this, these studies, but can I have the next slide, please? So this is a basic timeline of when most of the pediatric trials that we've been discussing have been done. And it was sort of the golden age of pediatric hypertension trials from 1998 to about 2012. And that was sort of in the early days of pediatric exclusivity. And there were roughly, I could find about 30 total studies done in pediatric hypertension, but about 18 randomized controlled trials. But here are some of the pivotal ones that we've been discussing that have been done over that period. Haven't really been that many since this time. This is, we've discussed the different trial designs, which goes into a lot of how interpretable these trials were. As discussed before, most of these studies were type C design, but type A shows a dose ranging study with a placebo arm, which is difficult for IRBs and patients to accept. B, but it is the most straightforward design. B is does not have the placebo arm, but the problem with B is that if the doses aren't selected correctly, it may not be interpretable. Most of these studies were done as type C designs using 3-dose studies like B, but then having a randomized withdrawal period. And the benefit of this type of trial design is that it is interpretable after the randomized withdrawal. And then the type D design is a titration to the maximal dose, which was done in very few studies. Next slide. So here is just a list of the basically most of the 16 drugs that have been approved for children. Of these, you can see that only 12 of the drugs had showed a dose response. And we've alluded to some of those problems, why the trials were negative, not showing a dose response before, but there have been issues with trial design, with how it was analyzed, how it was not dosed according to weight, issues with recruitment, various things. So, but in all, like about 3,500 children have been enrolled in these studies, and has led to only about 12 positive trials showing a dose response, 16 approved for children overall, and only 3 for younger children. So the purpose of this panel is to look at these studies and with our panelists and the audience discuss what are the lessons learned in all these studies, both positive and negative, and how would you design future trials optimally, to especially if we're looking at difficult to recruit populations such as young children. So should we start out with, let's see, I know Dr. Flynn has been involved in many of these studies. Dr. Flynn, can you tell us what you think were the major lessons learned and what your optimal trial design would be moving forward if you could redo everything?
Well, that's a tall order, but I think, like, for example, if we went back to look at amlodipine or another calcium channel blocker again, I think it would be important to look at the existing PK data to be able to decide about, dosing ranges and how to stratify between maybe different age groups or different weight groups of children. I think you could do more with trial design A, which Can you go back to that slide? Because didn't the enalapril and lisinopril and losartan did low, medium, and high, but I can't remember if they had a
Yeah.
placebo with. But I can't remember if you had a placebo withdrawal in those studies. I thought not, but I could be wrong. At any rate, I think we would ideally, I think we would have some PK data to help inform trial design. And yeah, they use design to see. Okay. And in terms of recruitment, the younger age group is a problem, as you've said. So clearly, you want a trial design that would either have no placebo arm or a very limited exposure to placebo. I think the other aspect of studying, these drugs is standardized blood pressure measurements. One of the things that was done by the sponsor in the amlodipine trials, that every site got the same model of Dynamap and education and how to use it. And that ensured sort of comparability of, quality of blood pressure measurement across sites. That was not done for many of the other trials. And I think, it would be something else to consider in designing a trial. How we're measuring blood pressure, is it the same across all sites? And then finally, the entry criteria. We talked earlier about how the enalapril, lisinopril, and losartan trials relied on diastolic blood pressure as an entry criteria, whereas the typical approval criteria in most antihypertensive medication trials is on systolic blood pressure. So sorting that out to enable design of a trial that will optimize the ability to show a response. And then finally, I think when the open label extensions were added for safety monitoring, I think that would be essential as well. Even, 1 year is relatively short, but it gives you a wealth of more data compared to the, shorter trials. So I'm not sure I covered everything, but I'm going to stop there.
No, that's excellent. Thank you. So what I'm hearing is more consistent primary endpoints and recruitment strategies and measurements with the blood pressure oscillometric versus whatever across studies, and then weight-based dosing. Okay, Dr. Bengus, do you have anything to add about what you think other things that we should note, lessons from these trials, and how you would modify future trials moving forward?
Hello, everyone. Just saying, I would like to say that I appreciate very much the previous presentation where there was this analysis presented about candesartan and valsartan. And I think that, the findings that were found there can inform the other ARBs. So I think that that's already a step forward for potentially other ARBs that may consider to be approved.
Okay. Any specific changes to trial design for trials moving forward, particularly if we were to study something in a younger age population?
I wonder whether it would make sense to have more similarities in endpoints that we are using, and also in the duration of treatment for the primary endpoint. I don't know exactly what was the similarity, what were the similarities or the differences in between these studies, for instance, that are showing here in terms of the primary endpoint and the time point when the primary endpoint was assessed.
Yeah, I think it was quite variable, and some measured changes in systolic blood pressure, and some measured changes in diastolic blood pressure. So that's an important point.
Yeah, would it be worth considering, more standardized approach for primary endpoint in younger population, perhaps?
Yeah, that's a good point. Okay, Dr. Ismat, same question. What's your wish list for future trials, and what have we learned from these studies, both positive and negative?
I think from Dr. Bengus's point, the commonality of endpoint and commonality of duration make a difference. And then the next step above that would be, would there be ways of pooling placebo patients across studies? Because if you have a common endpoint, you have common duration and similar design, that may be a possibility. Of course, no one company would like to take that burden on, but that might be something that FDA may be able to help with, or with academia may be able to help with. The one thing that actually I look at when I look at this set of studies is, for me, someone who's used to working in rare diseases, this is actually a lot of patients, in my opinion. This is actually a lot of patients. I'm actually even impressed by the number of patients. And so, and the other question that I had, and maybe this is a question that the clinicians can answer, it puzzles me a little bit, the degree to which there may be a reluctance to participate in studies in which you are likely or highly more likely to be a placebo participant, in part because, again, hypertension we think of as something that, accumulates problems over time. And if you have, 12-week study, 16-week study, something like that, to me, it doesn't seem like that big a deal to be a placebo over that time, especially with the opportunity perhaps to be in an open label extension afterwards. And so I wonder to what degree, patients or families have expressed concerns around this placebo, and to what degree maybe are those real concerns? Are they a real barrier, or is it just something that maybe we discussed in the earlier session?
Okay, Dr. Brady, do you want to answer that? You've been involved in a lot of hypertension studies. Is there a general reluctance to enroll kids when they will be exposed to a washout period where they won't be on their standard medications and then maybe randomized to placebo?
So I've not personally enrolled participants in a trial in which there is a medication, so I can't necessarily speak to willingness to contribute in that way. But I can share that, clinically, many families do have reluctance to start a medication in which the safety is unclear in the patient population. And so I think that this is something that, and not just short term, I mean, just yesterday in clinic, like, what are the long-term implications of my child being on this medication? Those are questions that family members have, and contribute to their reluctance to even initiate a medication. And so I don't know that that's something that can be studied necessarily, and it's not necessarily what we're talking about right here. But extrapolating from that, I do think that if the way a study is delivered to a family, since many of them would prefer not to be on a medication, and so, that they might be open to being on a washout and being on a placebo to see, does their kid even need to be on this medicine? And some families are very keen to help improve the knowledge of these medications, because their child is going to likely need to be on something for a long term. So I think a lot of it personally can be improved by optimized communication and talking with families about what's important to them in trial design. And I think there are some investigators that are looking, in fact, on how to best do that. So I'm sorry I'm not fully answering your question, but that's how I kind of think about it and approach it.
Okay, thanks. So going back to Dr. Ismat's point about these are a lot of patients that are enrolled, and I was really impressed by the elegance of looking at where you presented the candesartan and the valsartan by looking at the raw data from FDA. Is there, I mean, an industry sponsor would probably not want to do some big meta-analysis of these, but is it possible for FDA to look at not really children less than 6 here, but certainly prepubertal versus postpubertal and extrapolate backwards? Is that a way to look at some of the lessons learned if you can look at the raw data of these trials? Okay, we can discuss that later. All right, and then, because I think there are a lot of data here, and there is probably some things that can be extrapolated based on size and weight in these various studies and exposure. Okay, so are there other things that we can extrapolate from these data other than dose response? What do you think about safety, Dr. Ismat?
So just as an aside, before we get to that question, I think one thing that we might think about is I think a lot of the studies may be trying to attempt too much in one study. And if there's some degree, can we separate PK? Can we separate dose response? Can we separate overall efficacy? And so that if we have these little chunks, they may be easier to swallow from the perspective of a patient. I can certainly imagine that a patient signing up for an efficacy study where there's a lot of PK to be done, that can be extremely irritating. And so to the degree to which some of that might be separated, and we might be more willing to have, one.
So another question to the panelists, in terms of these trials, they're all relatively short-term, and most of the trials have included a long-term extension, and especially in younger children, it's probably more important to have a long-term extension. But as clinicians, what do you think is long enough? Or, and what kind of testing should be done? Dr. Brady?
Yeah, sure. So I think that's a great question. I think it really depends on what outcome you're most interested in, right? If you're interested in blood pressure lowering and consistency of that lowering, I would say at least 4 months. I'm just kind of off the cuff here because you want to make sure that it not only brings the blood pressure down, but that the patient is able to tolerate it and doesn't manifesting side effects, and whatever the dosing schedule is something that they're able to stick with. And again, if they're, real life is happening,
Was it taking a long time? How time flies, I can't believe
some, Dee Wevider, you're unmuted. I don't know who you are.
I think I was interested when you told me that one time your psychic told you that you will be something like
Perhaps the organizer.
I didn't have my coffee sitting next to me.
Mute this person.
kidney, relative to kidney, and you came in.
Is somebody, it's hard to do on Zoom.
Okay.
Oh, okay. I forgot what I said. Oh, yes. So, so making sure that, again, some of what I'm addressing is a little bit implementation versus, the strict, but I do think it's important because at least we can see, are there side effects maybe that come out a little bit later on? And again, making sure that the blood pressure remains in the range, because honestly, when I'm seeing a patient, I usually give them at least 2 weeks to see what the dose effect is. And if that's the point at which you're, kind of around the time you're stopping the trial, you don't really know what the full efficacy is. And again, side effects are important. And some of these medicines, I think we put them, there are some of them are 3 or 4 times a day. And I mean, that's really hard to do. And so I think that can be helpful. Additionally, there are other outcomes that are important, such as looking at end organ damage, looking at LV mass index, and you certainly need at least 6 months to see any change if you're going to see anything at all. And that's another way to sort of get an indication as to good blood pressure control. So if that's an intended outcome or a desired outcome, I think you need at least 6 months, and it'd be interesting to see the cardiologist in the room if they agree, but that's my opinion.
Okay, thank you.
No, I think that the open label extension data is really, really important. But for me, the rub is the patient and family burden for visits, and to the degree things can be done at home. And I know at-home blood pressure monitoring is still kind of a little bit rough in children, but to the degree that can be utilized, I think would be really helpful for answering a lot of these long-term questions.
And I can add on to that, because I've given some thought on this idea of how to monitor blood pressure in a trial. And I do think as much as you can augment that with virtual visits in between, you'll be successful. And so, of course, I'm going to get back on my blood pressure device platform here, but we need devices that are accurate in the age group that we're interested in studying. And we need to make sure that the families are adequately trained to measure blood pressure. And in fact, I think any trial that is going to rely on home blood pressure monitoring needs to make sure that it's not only validated, but that it's actually accurate using the Target BP AMA guidelines to make sure it's accurate in the patient that they're looking at right then and there. And then I would encourage you to have them measure your blood pressure on the Zoom with them to make sure that they are actually doing it properly and that they're doing it in replicate.
Okay. Let's see, we've got a little bit of time left. Any last thoughts about monitoring, long-term extension, best trial design, how to incorporate PK and dosing ideally from our panelists? Yeah.
Maybe
for-
one question. Sorry.
No, I was going to try to get at Elisa's question in the chat about interpreting the safety data when you have, patients who may have comorbidities, and how do you determine whether it's from the disease or the drug? And that's, I think, very hard, but perhaps, that may speak towards the need for longer open label extensions to be able to better differentiate what's going on with their disease process versus how they're responding to the medicine. And as Tammy points out, you do need a longer trial design if you're going to look at target organs. When we did the valsartan study, we looked at head circumference, but it was only a year. So, there was, to look at something like growth or development, you would need longer than even a year. And to look at it more robustly, you would need a much longer, study period, but then that becomes impractical. So, I think that trying to decide what are the safety events that you really want to capture, the important ones for, that would inform recommendations for patient monitoring, and balance, between that and feasibility.
Okay, thank you.
I have more of a practical question. This is for Tammy, maybe Dr. Flynn. So the blood pressure guidelines provide, blood pressure parameters for patients down to 1 year of age. And so if you're talking about trials in these younger kids, how are clinicians defining blood pressure in these less than 1 year age group? And also how would you define like what's a response to treatment? You know what I mean? Like,
Well, Joseph wrote that paper too, so I don't know if you want to share. But there's a wonderful neonatal hypertension paper that has blood pressure norms up till age 1. So I don't know, Joseph, I'll let you talk about your own paper, but that's what I use.
Well, I think we actually need a whole nother FDA NIH workshop on infant blood pressure. So, we're not going to get at that in this meeting.
But there are very nice tables and ways in which they give you tools on how to measure blood pressure. But to his point, they're based on very small numbers of children who are very different. And so some of the kids in each month age category may be primarily, so there are, I think, 6 some studies, and some of them are really premature, some of them are not so premature, and they're all contributing data to this normative database, which is not nearly as robust as what we have for the AAP clinical guidelines. And if you notice, when you go from this neonatal hypertension paper with the norms kind of jump when you go from 12 months to the 1 year in the AAP guidelines, so it's not really contiguous, but it's the best that we have, and there is guidance there for that.
Okay. Okay, thank you, everyone.
Thank you so much, Jennifer, and to all the panelists. We're going to take a quick break. If you can come back at 3:10, so it will be slightly less than 10 minutes, but if you can come back at 3:10 so we can resume. Thank you. Thank you. Our next speaker is Maria Learoyd. She's a clinical pharmacology director and head of clinical PK and safety sciences at AstraZeneca. She's going to be presenting on dose prediction and modeling and simulation and providing an industry perspective. Maria is going to be presenting virtually, so we'll turn it over to you, Maria.
Oh, hi there. Thank you. Can I just check? Can you hear me okay first?
Yes.
Yeah, okay, thank you. So thank you for the introduction. So I'll be talking about how modeling and simulation support dose prediction and clinical study design in young children with hypertension, and with a particular focus on children who are under 6 years of age. And this presentation is going to be from an industry perspective. Next slide, please. Okay. So children under 6 remain one of the most challenging groups for dose selection. So as we've heard some of the other speakers mention, the substantial developmental changes occurring across this age range, particularly in hepatic liver enzymes, organ function and physiology. So quantitative information is essential to account for age-related changes in drug disposition for dose predictions. And then at the same time, PK studies are more constrained than in adults or older children, with very limited blood sampling, time points, volumes, and relatively small sample sizes. So to address these challenges, we need to use model-informed drug development approaches to account for ontogeny, to optimize the clinical study design, and then also to maximize the information that we can get from limited data. Next slide, please. Okay, so in practice, the pediatric dose prediction is iterative. So early dose predictions are often needed to support the PIP and IPSP submissions to health authorities, and that's before phase 3. So at that stage, the models are based on very limited data and have a number of assumptions. Once the phase 3 data are available, then and often those may include adolescents, we develop a population PK model, and this will quantify the effect of different covariates such as age, weight, and renal function on PK. And at that point, we will also assess how exposure relates to efficacy, safety, and pharmacodynamic biomarkers. So this helps us to define the metrics that are needed for efficacy extrapolation if it is feasible and in line with ICH E11, which was discussed earlier. Yeah, sorry, stay on the slide with the columns, please. Thank you. So as our understanding of the therapeutic window improves, the dose predictions are refined, and the modelling and simulation helps us to optimise the paediatric study design. And as data emerge during the paediatric study, typically starting in older children and then moving to younger age groups, the model can be updated further. And even after the study is complete, the dosing may still be refined to ensure that the exposure and, if appropriate, the biomarkers are within the target range and meet the predefined extrapolation criteria. So I'm now going to take you through the developmental changes in the hepatic enzymes, renal function and transporters that need to be accounted for in the dose prediction models. Next slide, please. Thank you. Okay, so small molecules are often metabolized, and the panel on the left shows that most major hepatic phase 1 cytochrome P450 or CYP enzymes have got low activity at birth, often well below adult levels, with maturation occurring across infancy and childhood. So the levels of some CYP may approach or exceed adult activity. The ontogeny for the CYP1A2 and CYP3A4 enzymes has been clinically validated. So this means that we've got greater confidence in model dose predictions for drugs that are metabolized through those pathways. So by comparison, the in vivo ontogeny of phase 2 UGT enzymes shown on the right is less well characterized, and that's largely based on in vitro data. So this is important to bear in mind when we consider the model uncertainty. Okay, next slide, please. So renal elimination is important for many drugs, including ACE inhibitors. And as we heard earlier, renal function matures rapidly, approaching adult levels at about 1 to 2 years of age. So models that predict eGFR in pediatric subjects based on their age and weight can be used to estimate clearance for renally eliminated drugs. However, they do not account for the ontogeny of active secretion and reabsorption via transporters. And the current understanding of transporter ontogeny is less well characterized. So again, and there's evidence of age-dependent maturation in both hepatic and renal transporters. So where transporters are important for disposition, their ontogeny should be considered in the dose prediction models, and you can run sensitivity analyses to evaluate their impact on drug exposure using the models. Thank you. So monoclonal antibodies are not yet a standard treatment for hypertension, but there are new therapies being evaluated. So for pediatric patients, developmental physiology can affect the distribution and the clearance in ways that are not fully explained by body weight alone. So there are changes in extravascular volume, endogenous IgG, FcRn expression, lymph flow, and then in some cases as well, target-mediated disposition could also influence MAB PK. And an FDA review of modelling for recently approved paediatric treatments has suggested that, especially in neonates and infants, clearance is better predicted when allometric scaling is combined with a maturation function to capture those changes. So taken together, these developmental changes highlight that we need to use quantitative modelling to integrate this age-dependent physiology into the paediatric dose predictions. Okay, thank you. So for the dose predictions, we primarily use two model types. So there's PBPK models and then also population PK models. So here we're just focusing on PBPK, and these are especially valuable early in development before we've got pediatric data available. So they provide a mechanistic view of how drugs distribute across tissues and organs, while also accounting for developmental physiology. So this is especially important in children who are less than 2 years old, where maturation has got a major impact on exposure. For example, in the first year of life, body weight typically triples, alongside major changes in CYP and UTT activity, as well as renal function. So importantly as well, PBPK models also allow us to evaluate dosing scenarios ahead of going into the clinic. And you can evaluate the impact of age and weight bands, formulations, and study design options. So they give a stronger basis for pediatric dose selection when we've got very limited clinical data. Next slide, please. So in contrast to PBPK models, population PK models are built from observed data. So these would typically use the adult and adolescent PK data collected across the phase 1 to phase 3 studies. And these models quantify the variability in PK and identify the key covariates such as age, weight, and renal function. And then they also link exposure to efficacy, safety, and PD to help us to define the exposure targets, which are used to support the pediatric dose selection and extrapolation. So in practice, population PK models are often used for renally cleared small molecules and monoclonal antibodies. And prior data and existing population PK models from similar treatment modalities can also be leveraged for new medicines. So once pediatric studies begin, these models become especially valuable. So they can be updated with emerging sparse PK sampling data to refine the dose and exposure predictions across the population. And then modeling and simulation can also help us to optimize the clinical study design and maximize how informative the data are from the clinic. Thank you. So I'm just going to move on to practical aspects and then how we can use modeling to help with the study design and planning. So this slide highlights an important constraint where the amount of blood that we can collect is very limited. So the blood sampling needs to include PK assessments, immunogenicity for monoclonal antibodies, safety chemistry, and hematology. So in the EU, the guidance is framed around using total blood volume with limits for both the single visits and cumulative sampling, whereas in the US, there isn't a single universal standard. So the acceptable limits are typically determined through local institutional review boards and ethics reviews. So what is feasible depends on the age, weight, visit frequency, and the regional requirements. And you can see in the example on the right how constrained those volumes can be in practice. So pediatric sampling schedules need to be highly focused, with each sample clearly justified by its scientific value and balanced against the patient burden. Okay, thank you. So in adults, intensive PK sampling provides a complete exposure profile, but as pediatric sampling must not exceed the maximum allowable blood loss limits, it needs to be optimized to capture the key phases. So as a real-world example, we recently conducted clinical trial simulations for a pediatric phase 1 study where we incorporated prior adult data and a proposed sparse sampling schedule into hundreds of virtual trials. And these simulations showed that the PK parameters, clearance and volume of distribution, could be estimated with a high degree of precision with cohorts of 12 children per weight band. And the simulated PK parameter data set was then also used to statistically assess whether the PK extrapolation targets were going to be achieved for a range of sample sizes. And then for younger children, for example, from 1 to 3 years, where recruitment is challenging and the sample size might not be statistically sized. In these cases, observed data we would suggest could be supplemented by modelling and simulation to support dose justification. And then finally, just a practical solution as well. So PK sampling can also be staggered, where you have a proportion of patients providing sampling for specific time points, where others provide samples for alternative time points. And then these data are then pulled into the population model to create a complete profile. Next slide, please. And there are further practical strategies that we can use to minimize the blood sample volume. So we can use micro-sampling like VAMs or dried blood spots, which drastically reduce the blood volume required. And I think if the methods are fully validated, there is actually potential for those to be used at home as well. The bioanalytical assay can be developed to utilize a very small sample volume, as low as tens of microliters. The sample times for PK and safety assessments can be coordinated to reduce the number of visits and needle stick interventions. And then also we just need to consider logistics. So with drawing so little blood, we may not have any backup samples. So that means we should control the logistics and do, separate sample shipments so we don't have any catastrophic loss of samples and data. Okay. So overall, so from an industry perspective, the pediatric dose selection, particularly in younger children, is a model-informed dose justification exercise. And the goal is to support extrapolation from adults and adolescents to pediatric patients or younger children with important developmental differences in physiology and drug disposition. So PBPK and population PK modelling play complementary roles in selecting and refining the dose as pediatric PK, PD, safety and efficacy data emerge. So to be successful, these approaches must be paired with a feasible pediatric study design, including small sample sizes, sparse sampling, low volume assays, and trial simulation. So overall, robust pediatric dose justification depends on integrating modelling, extrapolation, and practical study design to support evidence-based dosing recommendations. Okay, thank you.
Thank you so much, Maria, for that talk. I know it's late in the evening for you, so we really appreciate your flexibility to join us virtually to give this talk. Our next speaker is Christine Ferrara-Cook. She's the Executive Director for Diabetes Clinical Design at Eli Lilly, and her talk will be on innovative trial designs and leveraging existing data. Thank you, Chris.
Hi, everyone. Well, thank you for the opportunity to come today and to talk about something that's near and dear to my heart, which is pediatrics, and then also how do we design clinical trials that are not only technically efficacious, but also optimized for the patient population under consideration, and really thinking about the patient not only as the child, but also the family unit that has to have the buy-in and really think about the operational success of these trials. So I think I'm preaching to the choir, but I just wanted to put this up here on the complexity of pediatric population in totality and looking at these just kind of arbitrary ages that we think about typically. And as you get younger, they're increasingly distinct from an adult population or your reference population. There's less historic precedence, more inter and intra-patient variability, and then increasing operational challenges that I think we've talked about throughout the day. So not easy studies to design, not easy drugs to get approved. So I'm going to talk today about two different aspects. One about the generation of the data. So the what is the disease? What is the endpoint? What is the response to treatment? Thinking really about the disease and the indication. And we've talked about this throughout the day. Again, does the disease exist across all age groups? Is the pathophysiology the same? Does the pathophysiology change during development? I'm an endocrinologist, and so I think puberty is like my nemesis. It messes up all of my biomarkers. So I'm interested to hear about you from a hypertension's perspective if you struggle with some of the same things. We've talked about response to treatment and endpoints. Are they the same as in adults? But then really the how. And this is where the design again comes into play, and making sure that the design is set up for technical and operational success. And those are not always the same thing. I am a basic scientist as well, and so I can design a beautiful experiment that would be great in a mouse study that is gonna just fall on its face when I try to get patients and their parents involved in a study. So those are things to really be thinking front of mind when you're talking about a design. I'm going to go through this talk on some of the most common examples of novel trial design, and really with a focus on how can we use existing data. And existing data is going to have a spectrum of historical data, real world data, data you're currently generating, but this is certainly not exhaustive. So before we get into that, I think I am not a statistician. I do play one on TV. I want to just talk about the difference between traditional power and then Bayesian probability of success. And these, again, are not things that you have to do one or the other, but they are two complementary ways to look at how do you set up a trial to give you the data that you want. I think we're very familiar with traditional data or traditional power, where you're looking at the ability to reject your null hypothesis, and that's all your alpha. That's looking at a certain effect size that you have decided is clinically relevant. On the flip side, there is the Bayesian probability of statistical success. And so that is using existing data, or what I know about this disease or about this mechanism of action. What is my confidence and or uncertainty about getting this result that I have powered my study for with this design? And this is a probability of success takes together the uncertainty. It includes external data, but also can be updated as you are generating your own data, depending on how you're pooling that pre-existing data. And you can update your priors, update your probability of success. And the key thing is that you may have some discordance. You may have powered a study to very appropriately reject the null hypothesis, but still have low confidence in the ability to be successful. That could be because your diseases are very different in, for example, the neonatal versus an adult study. The mechanism of action, you're uncertain on whether it's going to work there. You're looking at a subpopulation of very specific people with hypertension that may or may not reflect the existing data that you have. So I'm going to now go through a couple of examples. Again, these are certainly not exhaustive, but they're ones that we think about quite often in clinical design as ways to get around some of the challenges that we're seeing with the diseases that we're trying to tackle. And here with pediatric hypertension, one of the big limitations is that there's such a time lag from when adult drugs get approved to when exactly the pediatric drugs can get approved. And this leads to off-label use of medications because there is just this time lag. So adaptive trials are a way to reduce some of the white space and use existing data from a phase that precedes the next. Here I'm showing some of the example I'm using here is phase 2/phase 3, but you can think about these in various different ways, a phase 2A, 2B, a lead-in section where you're trying to establish and validate a PK exposure prior to going into a larger second phase. The first is traditional separate studies. So you do have time and space that is between your two phases. So this is your standard 2-phase trial. What I'm showing in the hash lines are data that is included in a confirmatory analysis or a confirmatory phase versus those that are not hashed, or the solid ones which are not incorporated and included. And you can see very clearly that in this standard 2-phase trial, or 2 separate trials, these are not included or incorporated. As we get into some of the adaptive trial design, you can have operationally seamless ones. This here, the data sets from the 2 separate phases are not incorporated into one final data set, you retain flexibility between the two phases. So you can update some of your dosing selection, you can update dosing arms, but you do have a main efficiency of having administrative time savings. This is all in theory under a similar protocol, similar submissions, and this does streamline some of the time aspect of it. The inferentially seamless ones have higher efficiencies and they sound really great because you are able, especially in rare disease, you're able to condense and include both phases and the data from both phases in your final analysis. But one of the things that is really important here is that you are also already writing the rules to the game. So you are essentially writing before you even start and planning before you even start. How am I going to transition from the first phase to the second phase based on the data? And I call it kind of writing the rules to the board game, and then you give it to your sibling and you have hands off. And so do you have enough information at the beginning to give it to your sibling who may or may not, you know, he's going to read what you wrote. And that requires a lot of upfront work. We've talked about control arms quite a bit, and I think we've hit on it a little bit with the placebo arms, but I want to talk about control arms a little bit more agnostically. And control arms can certainly be placebo arms, but they also can be active control arms. So I also want to keep that in mind here. There's various different ways that this can be done. You can think about having control arms that are purely external. And this would be studies which have no concurrent control arms. So the entire comparator is from historic or real world data. But I think now that we're in this area of improved technology and data capture and electronic medical records, we can really start to think about what is this external control arm? Do we have external control arms where we have patient level data in this real world land that we live in, where these external control arms can be much more robust and leveraged much more in our trials? There are hybrid control arms where you are using some of the existing data that sits out there in the wild while you are still retaining a reduced sample size within your current trial to allow for some combination and even some sensitivity analyses to ensure that your internal control arm is similar to the reference that you are using externally. And then there are entirely synthetic control arms. And these are ones that certainly with AI now coming up on the horizon, you can really start to get creative about how can I match my individuals that are in my trial with some appropriate controls. And that's one of the aspects also about Bayesian modeling and Bayesian statistical significance is you also can look at various aspects of your external control arms or your external knowledge. It doesn't necessarily just have to be, oh, here's the endpoint, but you can look at the variability, the standard deviation, the inter-person variability, changes over time. I did want to talk a little bit about single-arm studies just to be a little bit thought-provoking here, and hopefully we get a little bit into this more in the panel discussion. But are there pathways where you can really truly have single-arm studies in pediatrics? And I think we've seen from the previous talks that some of these hypertension trials, the treatment duration is very short. We're talking about between 4 weeks, 6 weeks. And so do you have the ability to have single-arm studies? Now, traditionally, most of there have been drugs that are approved with single-arm studies. Most of them do use a reference to some sort of natural history or a baseline control period. And these are really the strongest arguments are for those rare diseases where there's objective endpoints and a large effect size that is expected. It's less, you have less confidence with ones where there are subjective endpoints, potentially high placebo responses, or an unstable disease trajectory where the simple aspect of time can dramatically change something without necessarily having that be attributed to your drug that you are investigating. But at the end of the day, just early engagement with regulatory agencies and having very pre-specified objectives and decision criteria to make sure that everybody is aligned before you engage on these is essential. Master protocols, I did want to put this up here just kind of as a crash course on the various different types of master protocols. I think people use master protocols pretty loosely without understanding that there are various types. So there's basket trials where you have a single drug tested across multiple diseases, an umbrella trial where multiple drugs are tested within a single disease, and then platform trials that are kind of all of the previous ones. What are nice about these trials is that there are operational efficiencies, but there also are opportunities to leverage control arms across the different ISAs that sit under a master protocol. Right now, it is most often done when there are concurrent control arms, but even further as we get along, is there an opportunity to use non-concurrent control arms and really reduce the exposure of those pediatric patients to a placebo or to a control arm. And I think this also is going to become more relevant in pediatric hypertension, where there's often a combination approach. You're taking people that are pre-treated on something and then adding therapies. So where is opportunity here within masters to take these monotherapy arms and potentially borrow them as you're investigating some combination therapies, or I heard before about like co-formulations, which I love. And then lastly, I'll end on this. And this, you know, again, I'm a pediatrician. I now am a mom of 2 toddlers. And this probably should have been my leading slide. But one of the things that I didn't appreciate when I was certainly in practice and not a mom is that it is the family unit that we're talking about here. And so you do have not only infants and children who can't consent for themselves, but also an entire family that you're thinking about uprooting and bringing to clinic and asking for various forms of informed consent or PROs by proxy. So how do we really think about making this feasible, not only from a design perspective and ensuring that we get the data that we need, but also making sure that operationally we can execute them? So I've put in some of the ways that many of them have already been discussed today. But one thing I really want to put a nod into, and something that we on design really try to do forward thinking, is get interactions with patients, caregivers, providers early in the design process and often throughout the design process. So again, we are ensuring that our design is not only going to be technically successful if we get all of the data we want, but actually be feasible so that we can actually generate this data in a way that is going to have people stay with us until the end. So with that, I'm going to end.
Wonderful. Thank you so much, Chris. I'd like to invite Lynn Yao, who's going to moderate the next panel discussion, in addition to the many panelists that we have both in person and virtually. Lynn Yao is the Director for the Division of Pediatrics and Maternal Health at the FDA.
All right, we'll get our panelists up here, and we have some that are virtual, so we'll make sure we get everybody assembled. Let me just make sure I have the list here. Yeah, here we go. Yeah, Jennifer, come on down. And we have Monica. Is Josh coming back on? Maria, great. Okay. We have a big panel. This is a tough time of day, but it's, I think, probably the most important panel we have. of the day personally. But the idea is that we're going to bring everything together that we've heard today. And I really hope that at the end of this session, we'll have a good understanding of those very first questions that we posed to everybody at the beginning of the day. So I'm going to go slightly off script a little bit. And I'm going to ask, from your perspectives, wherever you're sitting from, whether you're in Seattle or right next to me, I want to know, and I could ask it one at a time, or I could give each of you a chance to opine on each of these. But we heard a lot about the ABCD, paradigm of treatment, right? ACEs and ARBs, beta blockers, calcium channel blockers, diuretics, and then everything else. And what I want to know is for probably the top three, when we say ACE and ARB, calcium channel blockers, diuretics, beta blockers, maybe we'll say 4. What are the gaps in knowledge for these drugs, these classes of drugs, in patients less than 6 years of age? That's the question that I have for all of you. What gaps in knowledge do we have in understanding how these drugs work in the youngest population? And who do I want to pick on first? I want to pick on Tammy because she's smiling at me.
That'll teach me. Well, today these talks have been phenomenal, and I just want to say that I have the wheels are spinning. I have all these ideas and I want to probably talk to all of you. But one of the things that I was thinking about, and I think it might have been your presentation, looking at how these drugs perform in different age groups. But in nephrology, we always talk about how hard it is to estimate GFR in the first 2 years of life, right? Because you have glomerular maturation, and you don't see your adult GFR until you're at 2. And so it follows, and again, I'm not a pharmacologist, but in that first 2 years of life, that you may have different efficacy than in older children and certainly in adults. And so that's one thing that I think that we need to be mindful of. And how, as I was talking to someone else earlier, how some of these medications that we use might impact maturation of the kidney and other organs. And so there's limitations to the period of time at which you can use them perhaps, or maybe not. And so I think more information in that regard would be helpful. And then again, what the dosing, differences might be. And then the other thing, again, I know I keep talking about this, but I always come back to the practical knowledge and how it's going to help me with my patients. But, little babies eat all day long, right? And then they stop eating all day long, thankfully. And so if the drugs are working in a certain way with food, and now their feeding patterns have changed and they're getting their medicines at different times of day, reference to food, that, can really impact things. And it's definitely something that is different than an adult. So those are things I think would be helpful.
So let me press you a little bit, Tammy, and I'll have other people jump in. Feel free to jump in, any of you online or any of you in the room. So, do the drugs have different efficacy, particularly in the youngest population? I heard, can we be confident in finding the right dose? What about safety, right? Like, what are the differences in safety? So based on your collective knowledge, and Tammy, I know you've been doing this for a while, have you noticed, or have you experienced, or have you identified real differences in efficacy? That is, this calcium channel blocker works in a 6-year-old, but it doesn't work in a 2-year-old. Or this diuretic works in a 7-year-old, but it doesn't work in a 1-year-old. Have you seen evidence of that?
Yeah, well, so I can tell you, I've seen some patients respond better to different agents, and I've been really taken by Joyce Samuel's work of her N-of-1 trials, and I've impacted my practice in that way. Instead of adding on medicines, just stopping and starting a new one. And I don't know that I have systematic look back to see if it's a particular patient that responds in a way, other than the person in front of me that's responding in a way. But I think that would be really helpful. I do know that, in peds, we do a lot of off-label dosing. And in fact, just sitting here, I texted my friend who's a peds cardiologist. I'm like, what's the youngest age you use irbesartan? Because I love, anyway, I won't get into it, but I like to use that medication because I find personally in some kids that does work better, but some of that is, if you look at the half-life and you look at the fact that it doesn't get impacted by food and it doesn't have any drug interactions. And so it's one of my go-tos. But again, we're limited in infancy because I don't know how is it going to impact the kidney? What age can I start using it? So anyway, hopefully I answered.
Yeah, very helpful, very helpful. I'll move down the line. Sudharshan, what do you think from your perspective are the gaps in knowledge we have in these specific antihypertensive classes? Particularly, I'm going to leave off neonates because I'm going to ask that later, but in 1 to 6, so 1 month to 6 years.
So I'm a clinical pharmacologist. So wearing that hat, I just wanted to mainly comment on the treatment response. And in terms of the available data, we primarily have data only for one class of agents, that is angiotensin receptor blockers in pediatric patients less than 6 years of age. So, Dr. Yao, related to the question that you asked, I think there was a point that came up in the morning that the disease etiology being different in patients less than 6 years of age, being mostly secondary hypertension compared to primary hypertension in older kids. One thing I can say that this is also in the published reviews for both the drugs that have been approved, and valsartan and candesartan, that is the treatment response in CKD versus non-CKD subgroups were similar in those younger kids. So that's something that I just wanted to address because I think that came up in the morning session.
That's a really important point, Sudharshan. Thank you for bringing that up. So I think Sudharshan's point is that at least from an exposure response or dose response perspective, that for the ARBs, we have pretty good data that, again, our group at FDA have reviewed and said, okay, it looks pretty similar, less than 6. And this CKD, non-CKD review also suggests that even though we have different etiologies, the exposure response seems to be pretty similar across or between CKD and non-CKD. So those, I think, thank you for bringing that up. I think those are areas where we maybe have confidence that that gap in knowledge that is actually doesn't really exist for certain classes and for at least for the ARBs. And between that question we had about CKD and non-CKD and whether that difference in etiology might play a role, it appears not, at least in terms of response to treatment. Okay, great. Jennifer, please.
And so in terms of gaps in knowledge, there have been a lot of comparative effectiveness trials in adults, and there, to the best of my knowledge, there hasn't been any good comparative effectiveness studies in children. So we don't know in a kid, for example, who's never been exposed to anything, whether to start an ACE or an ARB or a calcium channel blocker. I mean, it's just physician preference, or sometimes I'll ask, oh, if mom has hypertension, I'll just like give the same drug as what she's given, just for to make it simpler. But we really don't know long-term comparative effectiveness. And the other thing that we don't know is some of these effects on neurocognitive effects, because I've seen kids come in and say, I'm more tired, I can't play sports as well. I was in clinic last week and one kid said, this amlodipine is making me feel, quote unquote, dead inside. And I didn't even know what he meant, but he said, I feel dead inside, so I'm not going to take it anymore. And so, and what dead inside meant is that he really couldn't concentrate as well, and he was fatigued. But, so there are these neurocognitive effects that are not really well fleshed out.
Great. I am fascinated. Sorry, I'm taking notes because I have to close today too. But I am fascinated by the idea of comparative efficacy. I think we all, if we've ever treated a patient, we're interested in comparative effectiveness. That's a really hard sell at FDA, honestly. In fact, I think that drug companies go to great lengths to not have their drugs compared to others. And in terms of that difference between scientific questions and regulatory questions, I think that Joseph brought up, it's a really good, an important distinction. So really good point. I'm not sure that we're going to be able to answer that question at FDA, but a really good point. And I think an important one clinically. Ricardo, please.
So I am a safety physician, but I'm a pediatric cardiologist by training as well. So I used to use in clinical practice a lot of those drugs. And in the majority of the patients, when we use those drugs, I see that they lower the blood pressure and they work. I see that the great gap being a safety physician is whether they are safe. Long-term safety, it was already said, we don't have really any data regarding long-term safety. And this is an important issue. Another thing that I think that is a gap from a safety perspective, it's the neonatal population. The neonatal population, we have nothing. And we use it empirically, but thinking about risk-benefit balance here, when you have to use those drugs in neonates, it's really important because they are really suffering from hypertension. So you have to weigh the benefit-risk balance here. And if you use it without any information regarding safety, because it's really needed. So I think that the gaps that I see regarding safety, those are the major ones.
Thank you. Christine, how about you?
Well, I am a pediatric endocrinologist by training, but I'll do my best. I think that we've talked a lot about the long-term outcomes, but one of the things I'm curious about is the disease course trajectory and how that changes if someone is diagnosed as an infant and a neonate. Does that stay consistent? And does the response to treatment stay consistent across the developmental phases that a child goes through? And again, I mentioned my favorite, puberty, but how much does someone really evolve in terms of their safety profile to a certain agent or the efficacy profile to a certain agent? And do we know that? So I think that's something that's on my mind, is kind of thinking about the development aspect.
Yeah, terrific. I mean, one of the points I think we've heard is that a lot of these kids who end up on antihypertensive treatments are going to be on it for decades. And so does the need for treatment, the dose, the type of drug, the efficacy of a particular class change as a child grows, right? So.
Well, and I'll add that this comes into the design play as well. Is there a certain biomarker or covariable that we need to be thinking about strategically, instead of looking at this as a kid with this age, but it also is going through this developmental phase that I need to think about stratifying or at least taking account.
Yeah, and I think I heard Josh talk about it, and certainly you're bringing it up. One of the things that we have learned over time at FDA with respect to biomarkers in pediatrics, one of the poorest biomarkers is age. Right. So age we use all the time, and we use it in a regulatory and legal context, but really doesn't mean anything. Is age really just a poor biomarker for that change in ontogeny of SIP? Really, that's what we're looking at. We're not looking at H2. We're just looking at when does SIP mature, right? But nevertheless, and I think to your point, drilling down at what changes in childhood developmentally over time that may affect blood pressure and the treatment of it is, I think, a good question. Thank you, Christine. So I'm going to go onto our virtual participants. I see Monica next. Monica, thoughts?
Yes, thank you very much.
Yeah, anything that you think there's a gap in knowledge of the classes of drugs we've talked about today.
Yeah, thank you very much. And just to mention, I'm a cardiologist, but with experience treating adult patients. So all the topics today was extremely fascinating, and I feel like I learned a lot about, pediatric and hypertension in kids. from what I heard today, I was wondering whether there is a gap in knowledge in this youngest population with regards to the combination therapy, and whether we know whether the combination of, smaller doses of two drugs like diuretic plus ACE and ARB, for instance, is better as compared to an ARB altogether long-term in terms of efficacy, but also safety and tolerability. And I'm asking this especially because in adults, normally, the standard of care is represented by rather two standard of care medications and not necessarily one single dose medication at a higher dose, but rather smaller doses of two combination of therapies, ACE and ARB plus a diuretic or a CCP plus a diuretic. So that's something that,
Monica, thank you for bringing that up. And I think Josh talked about that I think he was the one that brought it up, the idea that we don't have combination therapies. We don't use them that commonly. And, we at FDA haven't even really required those. And we are going to have to rethink that given, this question. So thank you for bringing that up. I'll do Maria next, please.
Okay. Yeah, so I was thinking more about the, the effects of chronic kidney disease on the PK of these drugs, because some of them are renally eliminated. And, for adults, normally you would do a full renal impairment trial where you have, assess the effects across the range of GFR. But obviously in children, it's difficult to do that. So you would have your, your model-based predictions with PBPK, but then how much data do you need to actually confirm those doses in children with chronic kidney disease?
Yeah, very good question. In fact, we had a whole workshop, what was it? 8 months ago now, on renal impairment in pediatrics and the effect on dosing across drugs. But yes, that's a very important point. Even beyond what Sudharshan is talking about, Sudharshan was just saying, okay, look, we looked at sort of the subgroups of CKD and non-CKD, and their exposure response was similar, but it wasn't based on, degree of CKD and how much in a situation in which we know a drug is renally cleared, you would expect that would have an effect on dosing. But yes, a very good point, Maria. I'm going to go to Joseph next. And you've had a lot of work today, so I really appreciate you hanging in there and being a part of these panels.
It's okay. If you could share my view of the outdoors right now, you would understand why this is actually fine. Anyway, I've shared some photos with other people. You can ask Tammy. But anyway, so I have a number of thoughts on these points. I really was impressed by the analysis of the data for candesartan and valsartan, the extrapolation data. But the problem is every other drug class, we don't have similar information. So we're not able to make that same kind of extrapolation. So there's a gap. I agree, the gap on combination therapy is really important because there's only been one trial of a combination product in pediatrics. And, do we need combination, products studied? I don't even know if they're studied in adults actually, but that may or may not be a gap. And then I think this whole question about, let's just call it somatic maturation and dosing and dose response is really interesting. I think anecdotally, I'm not alone probably in observing that for many young children, as they're growing rapidly, we're increasing the dose of the medication, whether it's lisinopril or amlodipine, to get the blood pressure under control. And then all of a sudden, the child gets to be, 2 or 4 or 6 years of age, and they're on the same dose for the next several years. And why is that? Is that just related to the fact that we're now sort of, their metabolism is slowed down, that we've hit some sort of saturation of of, you know, their different enzymes. And so, you know, I think, and there, the gap, I think that really is important is a lack of PK data on some of these drugs in younger children. And, you know, if we had that, we might be able to better understand the, you know, how to dose and could you predict differences in dose response based on, you know, the just PK. And you can do things like a sparse sampling population PK, like we did with amlodipine, for example, amongst patients who already are receiving the medication. And then the last part about comparing drugs to each other. So right, there's only been one trial that compared two antihypertensives. I think it was valsartan and enalapril, or was it candesartan? There was one ARB versus an ACE inhibitor trial in kids with chronic kidney disease. And then we did do a real world comparative effectiveness trial of RAS blockade versus calcium channel blockers in children with chronic kidney disease. But that is sort of, you know, not necessarily related to what we're talking about today. So I'll stop there.
That's terrific. Thank you. Thank you, Joseph. And is it fair, Elisa, to ask you? I'm going to ask you, where do you think the gaps in knowledge are?
Yeah, and well, I think that's a great question to say, is it fair to ask me? I was thinking as you asked the question, I was thinking, I am not a pediatric nephrologist. I'm not pediatric. I don't, have experience with sort of real world treating these children. But I do just want to say that, you frame this as sort of, and as you should, Lynn, asking sort of where the gaps in the knowledge are. But having listened to the excellent presentations this morning, really the excellent discussions thus far, I have been impressed by how far you have come and how much data you actually have. I know we can focus on how little data there are, but because of that golden age, you were able to do a number of randomized controlled trials. And as I understand, unless I misunderstood the talks, you did generally see that things that worked in adults worked in your older kids and in some data as well in the younger kids. Granted, maybe it's not across as many pharmacologic classes as you want, but I think the data were reassuring. I also thought it was really reassuring to see, that for some of your intermediate markers, on the way to cardiovascular outcomes, that you're seeing these relationships, at least in the older children, in terms of seeing LVH and these other things. And I also just want to sort of emphasize or note that, we've had a number, as others have noted, trials in adults that have shown us that targeting lower versus higher blood pressure, leads to good outcomes. And I think that should maybe make us more confident that even though there are a lot of gaps in the data, that this is causation here, that the high blood pressure, even though we may not know exactly how to define it in kids all the time, that may not be so critical that we don't know the exact thresholds, because across a lot of different thresholds, higher is generally worse than lower. And again, maybe we're really targeting a really common underlying biologic process. So I think you asked about gaps, and I guess maybe I'm saying more to yes, gaps, but boy, there's a whole lot that you can leverage, I think, going forward in terms of how you think about designing these trials and really what data are needed to answer at least some of the questions.
Thank you, Lisa. And I swear I did not plant her in that position. But to Elisa's point, because this is going to lead into the next question that I'm going to ask all of you. We've heard everybody stated what they feel like are sort of some of the remaining gaps in knowledge. But I think we also just heard from Elisa and from many of the talks today, that list of, Ricardo, you said, you do rare diseases, but that's a lot of patients. Or who was that? Was that Ricardo? Oh, it was, yeah, right. It was Fraz, right? That's a lot of patients. And that's a lot of patients with controlled data. That's controlled clinical trial data that suggests that most of the cases that the drugs that work in children also, I mean, drugs that work in adults also work in children. So with that basis, that large clinical trial database, knowing that kind of as a baseline and understanding the gaps that we still feel like we have. You know, we don't have great exposure response in some of these other trials. We do know that the drugs appear to work. We don't have great exposure response. We don't have comparative efficacy. We don't have treatments. We don't have comparisons across, we don't have necessarily all the long-term safety we'd like to have maybe on neurocognitive outcomes, you know, as children grow and develop over the course of many years. We don't necessarily know how dosing changes over time because we don't have that exposure response data. We have sort of cut points. We know it works at this age, but in a particular patient, how does that exposure response change? But we also know that even in those clinical trials that there appears to be similarity between non-CKD and CKD patients. We know that there's similarity of exposure response, and we know a lot of these clinical trials demonstrated effective or efficacy similar to the adults. So based on that information, right, sort of constructing, and what I'm making you guys do is construct an extrapolation argument, an extrapolation plan. I think if we're creating an extrapolation concept, what we're saying is we have controlled clinical trial data that suggests the diseases are similar because similar exposure, similar responses, similar efficacy between adults and children in these randomized clinical trials. We have some controlled data, or we have some data that suggests response to treatment is similar. We know in terms of drug pharmacology that the pharmacology is expected to be the same across a pediatric patient versus an adult. The difference is how is that drug metabolized? And that leads to, you know, we need dosing information. So based on that, based on all that information that I've just heard, and I think we've heard throughout the day, the question for all of you now is, what kind of study should be designed to address those gaps in knowledge and taking into account what we know to be similar across the clinical trial data that we already have? What kind of trial could we consider based on all that information? Would we and should we continue to do randomized withdrawal trials in patients less than 6 years of age? Should we consider a PD-PK approach? Should we consider a Bayesian approach? Do we need to leverage adult data and run a controlled trial still in children? Do we want something else? So it's a tough question, but that's the question I'm going to pose to all of you now. And I think I'm going to go, should I go in reverse order now, or should I? Actually, I'm going to take volunteers. That's probably fair. Does anybody want to raise their hand and answer that question first? Oh, come on. Joseph, please.
Okay, so here's what I want. I want PK studies in drugs for which we don't have data in young children. And I think you could recruit children receiving them off-label and do a population PK approach to help define the metabolism. Adding a PD, you know, pharmacodynamics to that would be only feasible if you know, if they weren't already on it, obviously. At least as far as I understand pharmacodynamics, but I may misunderstand it because I'm not a pharmacologist. So that's my first ask. My second ask is to include children down to the age of 6 in all trials of new antihypertensive drugs for which there may be use in children, and not have children be an afterthought. So those are my two asks.
So PK studies in children.
With the PK, you could then predict the dosing, right?
Yeah,
So,
for dosing. And that can be in children already on the treatment, is what you're saying.
yeah, I think so. I mean, we did that with amlodipine, and you can look at that paper and as a, it may not have been perfect, but it's sort of a place to start for that kind of work.
Okay, and I see Aliza and then Monica.
Yeah, and I'm gonna maybe ask questions that I give an answer here, because I did just want to understand, Dr. Flynn, your last response. So I think one issue that has been raised is for these kids, maybe below, and others should help me here, below 1, tell me the age, where you're getting some maturation, where there may also be more maturation of these systems, either as relates to metabolism or the kidney, or there could be because of sort of some of the concomitant CKD, other issues that you may think that you may run into more safety, et cetera, concerns. Can you speak to whether, when you just made your comment about, I'm happy with just PK, whether that's through the down to, I think Lynn didn't want us to talk about infants at this point, but can you tell us how low you're talking about?
Right, I guess down to 1 or 2. But I mean, obviously the younger you go, the more exceedingly rare the patients are going to be, which is going to make it harder. And I don't want to say that's all I would like. I mean, but I think if we're going to try to figure out, dosing information for a label, then you have to have the PK data to know how to craft that. So that's why I sort of started with that, as sort of where someplace maybe to begin, particularly for these drugs where we're using them and we don't have any trials at all.
I'm going to ask a clarifying question too about that. So I'm trying to envision a study, let's say even an opportunistic study, that a patient gets put on a drug, gets put on an antihypertensive, and we want to get the PK. And the PK will tell us the dose. The PK that we get from it will hopefully tell us how to dose the drug if we know the PK relative to the adult PK or an older child PK. But that PD response would be the blood pressure, right? How did it lower the blood pressure? And for that, do we need patients who haven't been treated? Do we need a trial in which we take patients who haven't been treated, who are diagnosed with hypertension, get put on the drug, and then collect the PD, which is how much did it lower and what was the dose that was used? Or do we have enough? Are there certain classes of drugs that we don't need that in because we know enough about it? We know it's going to lower the blood pressure. And all we really want to do is confirm the PK, as Joseph is saying, and make sure that we have the PK that supports the dose that we believe they have always or should be on. I want to make sure I clarify that fact because it wasn't clear in my head. Like, do we want trials in patients who haven't been treated yet? Or is it okay because there's so many kids out there who are already being treated with these classes of drugs to just confirm what PK we have on the doses we've always been giving them and compare that with older children or adults?
Yeah, I mean, Lynn, I think if in the best of all possible worlds, you would do a trial if it were, no barriers to recruitment, you could easily measure the blood pressure, and, families would be happy to put their children into a trial, and we could design the perfect trial. I think it would be great to have studies that on kids that have never been treated. But the reality is that there are not many. It's going to be, we're not in the perfect world. There's a lot of barriers we've talked about. And we do know that for most of these drugs, just from clinical experience, lower blood pressure. And there are other ways to get safety data, because there are, I'll just talk about PedsNet again, because, you know, you could use laboratory data and blood pressure data and medication dosing data that are discrete data fields in, you know, a database like PedsNet, where you I would think you could pull out some of that stuff on safety and so forth. That's how we did the comparative effectiveness for calcium channel blockers and RAS blockers in CKD patients. So, you know, I think, you know, you don't want the perfect to be the enemy of the good, right?
Sure.
So just to figure out at least where to begin, given where we are in terms of already using a lot of these drugs.
Very helpful. Monica, please. Oh, Monica, and then Elisa again.
Yes, thank you. I just wanted to say that I like very much Christine's presentation about the innovative trial designs. And I was thinking about this external control arm, or even bolder synthetic control arm, thinking of, you know, potentially smaller studies with smaller sample size, perhaps without placebo arm, just, you know, considering having or constructing an external control arm that could help you. So
Let me ask, but let me just push you on that point though, Monica, a little bit. If the issue is that we need a control of some type, right, then there is some question about the efficacy that we think would be answered by a controlled study. Do you think that there are gaps in knowledge that require the use of an external control? Because if we decide we go there, I think to Joseph's point, we are saying that we need de novo patients who have not been treated, who need to be in a controlled trial, and externally controlled. They'll all get treatment, but they'll be compared to an external control. So that also, and we're talking about, I hope we're talking about largely the population less than 6.
Yeah, exactly. I was just thinking in terms of gap. I was more thinking about, you know, how difficult it is to find these patients and construct the study all together. And I was thinking that it would be easier and more innovative to do something like that for a smaller sample size. Sorry.
Very good. Thank you. Thank you, Monica. Elisa, please.
Yeah, maybe just a comment on that point, and then had a follow-up question. And I apologize, but you guys are such an exceptional group that it's just very helpful to, you know, to be able to tap into your expertise and understand, what you're thinking. Just, you know, in terms of external controls or synthetic controls, I think the challenge there is you really got to know really well what would happen to patients in terms of if they hadn't been treated. So what that alternative state would be. And I think for many settings, and certainly thinking to adults in hypertension, I can't necessarily tell you that. So I think sometimes, even though the designs are appealing, it can be hard to use them in some of our spaces. But that doesn't negate sort of, and one could also sort of, you know, it depends on the question you're asking. If you're trying to understand safety or something, maybe you have data that you could leverage, et cetera. But it's just sort of a general observation about sometimes using external controls. Typically, they work really well when you sort of have huge treatment effects and you really do understand sort of, you know, what would happen in the absence of an effective treatment. But just one other, I guess, question for the group was related to what extent do you think studies are needed to sort of test strategies for mitigating certain risks? So, you know, I think certain risks of these agents have been highlighted, you know, whether it be hyperkalemia, AKI. I mean, to what extent do you need a study to sort of say, hi, it's single arm, you know, everyone's getting drugged, but I specifically want to see whether, you know, if I evaluate or get labs at some frequency, you know, this is sort of how much of a issue I'll have with, you know, really bad hyperkalemia or really bad AKI. Can someone clarify whether you think you need data on that, or no, you think you understand how to monitor these patients to mitigate risks?
Ricardo, please.
Yes, I would like to talk about it. I think that this would be very valuable to understand the effects of the drug and the adverse events and serious adverse events that those patients could present, even don't have a placebo arm. I think that even if you are able to gather the data of the real world evidence and bring it to our reality and shows, oh, those patients are presenting more AKI events, or we have a lot of patients presenting AKI, although they are controlling the hypertension, it could help a little bit. But thinking about patients that are using real-world evidence data, I can say that it would be a little bit biased because only the patients that tolerate the medication that are using that will bring you the data. So having the single-arm study, understanding how many of those patients had to withdraw the medication due to adverse events, how many developed AKI, how many presented serious adverse events, from a safety perspective would help a lot, even without a placebo-controlled arm. I think that from a safety perspective, it would help.
Thank you. Tammy, please.
Yeah, I think understand. So that would be really helpful from multiple standpoints. I think it would help clinicians maybe be more standardized in terms of how they're screening, but also in communicating with families. You know, you're always talking about the risks and the side effects, and it can be really scary to hear, oh, I just need to check your labs in 2 weeks to make sure your potassium and your creatinine don't go up. But being able to have some hard data to explain that. And how frequently it actually occurs. But almost to answer a question that was posed earlier and thinking about what other things we would want to study in these trials, I think it would be really great just thinking again at what are the things that matter to families, because I can prescribe all I want, but if they don't want to take it, they're not going to take it, right? And if there's some concern, some perceived side effect or adverse effect that's going to happen, they may not take it. They may tell me they're taking it, but they may not take it. And so thinking about things that that's important to them. And I was just scribbling some things down. And these are softer outcomes, perhaps, and maybe would be more apt for some of those extended open label parts of the trials. But looking at school performance, you know, their cognition, absenteeism, their appetite for the younger ones. Are they irritable? Are they sleeping well? Is there any change in that? And because it really is the families. They're just the questions I get are, what is this going to do to them long term? And if you can tell them, maybe if you could answer that, that would be great. But if you could also tell them some of these other non-blood pressure lowering benefits, you might get a little bit more buy-in. Because at the end of the day, we want their blood pressure to be lower to protect their overall cardiovascular health long term. And again, to do that, you need to partner with them and get buy-in. And so those are some things that I think would be helpful. Again, I'm probably getting off topic for what we're looking at here, but I do think that would be helpful as we're building trials.
Christine, please.
I have a question for this group specifically with hypertension. I worked in rare disease and we're talking like 80 people in the whole galaxy with this. And I worked in a pediatric only disease. And is this pediatric hypertension group with registries and getting just, you know, all etiologies, pathophysiologies, having electronic medical records, lab data, but then importantly, like patient-reported outcomes. I think something I hear a lot from parents is I just want my kid, even just the stigma. I don't want to go to the school nurse. So 3 times a day is a non-starter. Is there a registry effort within this indication in this space to generate that patient-level lab biomarker qualitative data to then be able to use that with a little bit more solidity?
Yeah, I mean, I'll let Tammy weigh in, but I don't know about hypertension per se, but in chronic kidney disease, yes.
Yeah, and I think particularly with peds nephrology, we're really good at registries. But again, maybe not in hypertension. And I forgot, thank you for asking this question because it made me want to bring something up because Joseph brought up PedsNet. I think, you know, it's really great to be efficient and use existing data. But again, I'm on my blood pressure measurement platform here. I would really hesitate to use medical record blood pressures in any way for this because I can tell you from my clinical standpoint, I've done studies myself where the triage blood pressures are often very different than standard of care blood pressures done by manual auscultation. And I shown that can be up to 20 to 30 millimeters of mercury off. So I would encourage, please do not use EMR data for blood pressure. Other things, labs, et cetera, absolutely, but not the blood pressure,
Or even
please.
just association of, use of blood pressure with certain, safety outcomes, right? Yeah. The Superman registry.
Superhero.
Superhero
Yes,
registry.
with Andrew South and Wake Forest.
Yeah.
That's right. That's right.
Yeah.
I forgot about that. How did I forget about that? Yes.
So I've heard a lot about, the idea of collecting safety information, but I still want to drill down. I'm still trying to get at what does a trial in less than 6-year-olds look like for these ABCD drugs? What does the trial look like? I heard at least Joseph support, PK and patients who are already being treated. That's the gap in knowledge. We'd like to make sure we have the right dose. I've heard it's lovely to have some additional safety. That would have to be longer term, and we'd have to consider registries or other means to collect that data. Is that what everybody believes is the type of trial that we should be doing now, less than 6? Aliza.
Yeah, and I'm going to push further because the other thing I thought I heard was we're trying to get data that are generalizable here. And it's also, we want this to be feasible. So in terms of my push, I guess what I'd like to understand as well is to what extent is this sort of a trial where you really are trying to do, and I'm going to use the term pragmatic, and maybe that's not the right term, but just one that really integrates easily into clinical practice where you're just, again, maybe the one thing you are doing is you're making sure everyone's using the same device and they're adequately trained on it. But that a lot of it is then just sort of following these patients for some time period in this sort of single arm trial, getting some PK data and being able to sort of, even though there's not a control, just to sort of say, hi, we're sort of seeing these number of patients who are able to stay on drug. And yeah, it looks like, even though it's not, again, controlled data, that the blood pressure actually was, within the range I wanted it to be. And it was just on this agent, or it was on these doses of this agent. And to what extent are people looking for a trial that's really one that's integrated into clinical practice and has those types of elements?
Yeah, please, Tammy.
Yeah, I mean, I think so again, as I mentioned, I like to be very practical when I'm designing studies and thinking about things. So absolutely, I think the more that you can integrate what you're doing into clinical care, the easier it is to enroll patients, engage patients, because you already have that rapport with the patient, and not only they may help themselves, but they'll be able to help others. And so it's a lot easier to engage patients. But I think you have to be also very thoughtful about like, to your point, at the end of the day, what question am I trying to answer, right? Am I trying to answer, how am I going to dose this? What are the side effects? And how much does this lower blood pressure? Or am I trying to say, am I going to be able to get people in goal with this and in a real world setting? And I think the first question we still don't really have the answer to in that younger group. And I think once we have a little bit more data there, then implementing that in the real world to see, and some of that will be informed by, the side effects and the ability to lower blood pressure. One of the things I think that we all need to be thoughtful of in those real world trial, is that the importance of how long does it take to get the patient in control, right? Because patients are going to get frustrated. I keep coming back. The blood pressure is never in the range. You keep adding the medicine, and then that gets them in the real world to lose faith in what you're doing and maybe not be as adherent. But again, I think that those kinds of questions are separate. In my opinion, I think we're still sort of looking for the first question. And to Joseph's point, enrolling prevalent patients, I think, is easier than incident patients. And certainly if you're trying to enroll from your clinical practice, patients who already have the disease and are already on the medication, it's a lot easier to get buy-in and get them to contribute to the study because there's very little difference, so to speak, in what they're doing. And then if you want to do these real world or easier to implement monitoring by doing home blood pressure monitoring, and you teach them as a part of your clinical practice how to do the blood pressure measurement, and get, make sure the devices, not only are they giving you the data, but now you've given them a life tool that can help them in their own self-management long-term, which again helps with buy-in.
So I'm hearing that a study that evaluates PK, some short-term safety, and then using other sources for longer-term safety would be reasonable in this population, at least 1 to 6. Is that what I'm hearing? Does anybody disagree with that? Jennifer, please.
So I have tried to enroll a large number of kids personally in PK studies. I don't have any problem enrolling kids with heart disease who are in the hospital because they have an indwelling line and you can tell the parents, okay, we're going to just take some samples while your kid's in the hospital. It's not going to hurt, we can use scavenge samples, we can do all sorts of things. But when it comes to enrolling patients, which, in this case, we're talking about less than 6 now I understand that pop PK, smaller doses, that those smaller samples are all great. Nevertheless, families have to come back multiple times and more often than not have to get venipuncture, and they just don't want to do that, or else I can't enroll them. So I've stopped trying to do that, but so I think getting PK studies in kids in the NICU, or if somebody's in the hospital for some other reason, that can be done, but it's just a lot harder, in multi-center studies to have people to come back and have their toddler get stuck multiple times.
And that sort of goes to my last question. Do we have time? Okay. Sorry, Eliza, before you go to the next question, go ahead. I'm going to hold that thought though, Jennifer, because I think it's an important one.
Yeah, no, and it's actually just a follow-up question. To what extent this issue could be resolved by actually having people go to the hot child's home to actually, get a sample for PK, to address it that way. Is this the issue of actually having to come to the physician's office to the hospital, or is it even more than that?
I think it's the coming as well as wrestling down this screaming toddler to get blood. I mean, I wouldn't enroll my own child or grandchild in something like that. It's just very traumatic for the family. And so that's why I kind of wish that there was something else we could use, like saliva or tears or something like that. But, it just doesn't work out that way.
Tammy, please.
Yeah, so I completely hear that. But I think that we can perhaps learn from some of our other colleagues and other disease processes, because there is a way that they will bring their infants in for PK studies for different disease conditions. And I can tell you that in CKID, we were able to enroll people and to do these studies, to do iohexol, because the families were just so committed to learning more about the disease. If you think about it, if you're under 6 and you have hypertension, I mean, that's not like you're 13 and have hypertension, and it's not like you're 30 and you have hypertension. Those families are very concerned about why their child has hypertension, and I think you might be able to enroll them a little bit more easily and efficiently, like taking advantage of perhaps inpatient admissions for things that wouldn't impact the way that the PK studies. And then I had another thing that it's gone. Oof. Okay.
It's late. It's okay, Tammy. It's late. Minimize. Oh, please. Who was it?
Yeah, I think.
Oh, yeah, please.
I just wanted
Fraz.
to follow on your question and talk about adaptive experience. If you can combine blood spots or some of these other approaches at getting blood that are not involving venipuncture and being at home, not visiting, I really think that could probably. There are some people the business solution. For some people, the blood draws. Some people it's both. But there may be ways of kind of squeezing out information. And getting back to the question, I think that Dr. Flynn mentioned about patients on chronic therapy off-label, and sending, I don't know, the PK scientists will have to decide if there's any value on this. Send these families out cards and say, okay, you're on this chronic drug, just send us some blood spots, tell what time you took it roughly and what time they took the medicine, and then we'll make something of that data. Especially if they're on a steady state, maybe there's some value. And you don't necessarily have to know what the PD is because they're adequately treated to the extent that their physicians are happy with it, and then that will tell you at least something.
Thank you. And, the next question I was going to ask, you guys already answered. Oh, please, Sudharshan.
No, I just wanted to add another perspective to this discussion. As long as we are talking about PK characterization less than 6 years, but going down to maybe 1 or 2 years of age, we just need a reasonable precision on the PK parameter. So in terms of sample size, it's not a whole lot of patients that we are talking about. And I think it was touched upon in Maria's presentation too. So you actually maybe need maybe 10s or 20, patients in the range of 10 or 20 to get that reasonable precision on the PK parameter. So yeah, I just wanted to say that it's not a whole lot of patients we're talking about.
In fact, I think the talks that we've heard today, Maria, Sudharshan, the idea of how far we've come with using PBPK and POP-PK, that to obtain reasonable precision, we're not talking about 20 blood draws over, 7 weeks, people coming and visiting, weekly. It's really a couple of blood samples per patient, maybe a couple of times, and that we could get with a certain sample size, modeling and knowing sort of the variability in that drug, we could probably come up with pretty good characterization. Yeah. Christine.
I have something just to add to the whole feasibility thing. And Tammy, I don't know if you're just like exuding this or what, but the trust between the provider and the patient is so important. And so if it's 10 samples across the course of a year, if there's not that level of trust, it's still going to be astronomical versus being able to present that you're contributing to this pool of knowledge. And that, I think, you are just showing how engaged you are in your patients. And that's something we think about operationally when we're choosing sites, just for your traditional trials, is how do you really engage with investigators that are going to give that TLC?
I think that's a huge point. I, in fact, wrote it down, but, part of the reason why I think clinical trials don't go great in pediatrics are that there's a clinical trial group that kind of descends into a population. They're not the same people who they're seeing as providers every day. And so they're like, who are you? I don't know who you are. But, our pediatric community is, I think, small enough and coordinated enough that, if we said this is a coordinated effort that we want to do, we think we can do it. We have a few drugs that we want to get PK in prevalent patients. I think the nephrology community is already treating out there could reasonably understand that that's an important thing to do and get those patients enrolled.
Yeah, I think that's really key. It really is really important to have that trust and that rapport. And I've seen that in my studies when I've been the one that's recruiting versus my research coordinator who's, just to your point. And I remembered my other thought. So I really enjoyed your talk about sort of novel designs. And I think that I would love for all of us to really think outside the box. Like, instead of doing things the way we've always done them, are there ways to be more efficient? Do we really need to do, and this was touched upon, the traditional sample size calculations, which give you this monstrous numbers? Like, let's really think about efficient study designs so that when we are enrolling these patients that we're already concerned about numbers and burden, can we do that with fewer numbers and get the same answer? I think, there's someone at Children's National who is really thinking a lot about this, about these novel designs, and we need to engage more with those investigators.
Great, thank you. Can I ask one more question? How late are we? My timer says we're late. And also, Heather, do I send you my slides for the closing, or who do I send them to so I can get them projected up here? I haven't, because I'm just working on them.
Share screen.
What's that? Okay, because I think there's a way to share. I just have to get the email of the person to share them with. Adam? Okay, hold on. Let me see if I can do this right now. Adam, what's his last name? Stamper. Oh yeah, I have them. I'm going to share them with him right now. Okay. While Adam is going to load up the slides for the end of the day, the conclusion. Too big a topic, but I have to, if I don't get an answer or comments about you from this, I'll feel like I haven't done my job. Neonates, 0 drugs approved. What do we have to do? What do we have to do? 0 drugs approved. That's not okay. Is it too late in the day to ask that?
That's a happy hour workshop.
We meet at McGinty's in 20 minutes and talk about it? No. What do we need to do? Are the neonates so different that we can't do the same thing? Is that just off the table?
Well, Lynn, the problem is we're not even sure how to diagnose hypertension in neonates to start with. So, that's why I made the comment earlier that we need a separate workshop on hypertension in less than 1-year-olds.
Okay.
Starting with how do we collect normative data and then moving to treatment and so forth.
All right. I will have to accept that, Joseph. It's not what I wanted to hear, but I will accept it. Yeah, please, Carissa.
We say that kids have hypertension, so we aren't making a diagnosis of hypertension in their settings. As to the exact cutoff, I don't know. I would say too that one of the challenges though is that you're training up a neonatal population. So what are they in the hospital for? So you've got chronic hypertension, you've got chronic kidney disease, congenital heart surgery. So I think that just changes the milieu of the population that you're simply getting this information from. Because some of them are going to have kidney injury related to their bypass times. They're going to have, other factors, right? That are have genetic disorders that are hospitalized. So I think that's where it gets a little tricky. So we do in fact say neonates have hypertension, but there's just what's the exact number that we.
Right. And, and again, FDA is not in the business of encroaching on practice of medicine. We recognize, and I was treating neonates with, nifedipine pulled out of the needle from the capsule and asking, can you just give 1 milligram? So I did that. That was me. A hydralazine IV. I did not do the 1-to-1 conversion though. I knew that difference. But regardless, we know we're treating. It's just it feels very inadequate to have nothing approved. And if we're not there yet, we're not there yet. And I think everybody here has given us the idea for the follow-on workshop here, which is probably to address this issue in neonates. So yeah, Tammy, go ahead.
So maybe the answer then is while we're waiting for a better answer, maybe we do need to have a registry. So those of us who are doing this in the real world, if we're able to enter this in a registry and we can sort of follow, all right, well, I gave this kid with this diagnosis, this dose, and this is how the blood pressure changed, maybe that'll help us move the needle a little bit because you're just being efficient with knowledge or information that's
it's just right here.
Or not information, but clinical care.
Okay, step up.
You're getting a lot of hemodynamic variability, right? In fact, all of that. So, and then blood pressure varies from moment to moment.
Right.
So, it's not that it's impossible. It's just a lot of data.
Yeah. Yeah.
Have enough sample size to address, right? The number of variables in the model.
Yeah. Yeah. Shamir, go ahead.
It's harder or easier to study neonates? Easier because usually when you encounter them, they're hospitalized.
Right.
They're monitored, they're getting blood drawn, they're having blood pressure done regularly. Harder, they're complicated in the hospital. They've got a lot of things going on, some of which are contributing to the increase in the blood pressure, which may go away after they get better or go home and may not. Sometimes it's obvious why they have hypertension because you're consulted, you do an evaluation, they've got renal venous thrombo, any number of things that can occur to the hospital. So you have to recognize, like, I didn't see many neonates come to me in outpatient clinic that were discovered by the way blood pressure was elevated at, you know, before they left the hospital after a normal birth. It's always consulted for a sick neonate or complicated neonate in the hospital with developing blood pressure.
Yeah.
That may or may not be transient, but collect the data because we have the data. But at the same time, there's a lot of co-meaning factors that are contributing to it.
For sure. Yeah.
The other thing to consider though, some of these kids are going home with oral agents.
Right.
And this may be something, so after I said all that about registries, but if if they have an art line, which is an actual direct measurement of blood pressure, this might be something ripe for the EHR extraction.
Right.
So that's my
Right.
caveat.
The gold standard, I mean, our intra-arterial measurement, right, is one of the gold standards. So yeah. All right. I really appreciate everybody's hanging in there and allowing me to kind of go over time a little bit. And I also heard, I'm pretty sure I heard that all the people on the panel and maybe some of you in the audience are also going to agree to participate in the planning of the neonatal hypertension workshop. So thank you for your volunteering. Okay, so in the last 5 minutes or so, I'm just going to try and wrap up if I can. A very, very productive day. Thank you all. So in session 1, we talked about, and this is what set the stage here, right? That, you know, we know that drug development for antihypertensives is conducted in adults first. We are not doing de novo pediatric antihypertensive drug development. So it's a follow-on from adults, and therefore FDA does have the authority to require studies under certain circumstances. And even with that authority that we've had now for almost 30 years, we haven't done a great job. You can see here, as we've already heard, 61 drugs approved in adults, 16 approved in children, none in the neonates. Next slide. So, you know, what are we looking for today? What were we looking for today? You know, can we require new dosage forms? Yes, we can. We heard a lot about new dosage forms and how they can be used in this population less than 6. The opportunity is here. How do we fill the gaps in knowledge in patients less than 6 years of age? And I'll say today we focused on 1 to 6. We're not 1 month to 6 maybe even, but not the neonates. What are the studies that are needed? What are the studies that are feasible? How do we leverage extrapolation? What off-label information can be leveraged? And what study designs can be considered? And I think we covered all of that today. So next slide. So similarities and differences. I took a lot of notes here. I'm not going to go over everything. But what I heard, and I think Eliza sort of pointed this out, similar pathophysiology, similar biology, similar target damage. Measurement is the same. The norms are different, right? We heard, you know, static versus percentile based. Okay, that's different. Comorbidities probably less common in children. Etiologies are different, right? Treatment paradigms slightly different. We have fewer patients to study. And we have relied on a validated surrogate. That's blood pressure. We don't go for the clinical endpoint of stroke or heart failure, right? But in terms of consistent endpoints, we heard not necessarily always the same. Systolic blood pressure in adult trials, diastolic blood pressure, or systolic in some pediatric trials. Next slide. It's a lot of similarities. You know, endpoints we already talked about. And I think this is the point that Elisa made in all those controlled trials, that we saw efficacy in almost all the classes that we study, not necessarily in every trial, but in all classes. That antihypertensives lower blood pressure in all age groups, and maybe RAS blockade has a greater response in certain populations, like those with secondary hypertension, particularly in CKD. And then older hypertensive agents, we don't have a lot of good dosing information. Even though they're approved, we don't have a lot of good dosing information. A lot about off-label prescribing, and unfortunately, that's sort of the standard right now for children with hypertension. 70% were off-label. Neonatal was 100% off-label. That's really just, we have to do something about that. Not today, but we will do something about that. You know, there are gaps between what guidelines say and what FDA has approved, so we're trying to close that gap. And a lot of information now on newer dosage forms that I think are really the future. Mini tablets, pellets, powders that require less issues with palatability and probably less issues with what's the word? Stability, so shelf life. Next slide. Oh, even with that, you know, I just will say, even with that, when we don't have the right formulations, I think we heard very clearly that that's where we get into trouble. Compounding errors, mixing errors, administration errors. So, you know, what's the thing that we have to do? We have to think about how can a child take it, and then will a child take it? I love that. I love that, the way that was framed. Next slide. Okay, so what did we talk about in panel 1? I think the most significant unmet needs we heard were we need approved treatments less than 6 years of age. We need approved treatment options for patients, older children and adolescents who have hypertension and metabolic syndrome. We probably need to start thinking about new agents and fixed dose combinations for patients who have uncontrolled hypertension. We need longer acting medications and drugs that maybe don't have as many interactions. We need dosage forms that are suitable for all age groups, particularly neonates. We need potentially use of markers that we can leverage for comparative efficacy, excipients that are safe, both in terms of the type and amount in neonates, and then how do we assess the safety of excipients? Next slide. So what are the differences? So we heard about the differences, and I'm not going to go through all these because I think we actually got through a lot of how we can overcome these differences. But importantly, at the bottom, I think, and we talked about this at the end of the panel 1, like, do any of these differences preclude, like, including patients in a pediatric hypertension trial? Like, we wouldn't include CKD patients because they're so different. We have to include a different population. We said all comers. If a pediatric patient has hypertension, you could enroll them in a trial. So I think that was an important finding. No, we could include pediatric patients regardless of etaology or regardless of some of the other differences we noted. Next slide. And then, you know, what is the pharmacologic response? Is it expected to be similar? We have seen, and actually in the presentation that Sudharshan gave, we expect the exposures to be similar, and we've seen them to be similar, at least in one class. And neonates, again, I don't know what to say about them. We don't know. And then in terms of RAS, particularly in secondary and in CKD, there might be differences in terms of that population and non-CKD. What we heard was in terms of exposure response, we didn't see much difference. But this was the end of panel 1. And then were there any differences that precluded a population to be enrolled? And the answer was no. So next slide. So then we went into session 2. Dose response, as you heard, and I'm not going to go over what Sudharshan said, but basically for a couple of ARBs, we saw very nice exposure and dose response. It supports, you know, the acceptability of extrapolation, at least from an exposure response perspective. And of course, we have to consider safety. So what are the safety concerns? Age-dependent risks based on maturation, comorbidities, what is the underlying disease? Those are all considerations for the acceptability of safety extrapolation. Next slide. So in panel 2, you know, what did we learn? What have we learned from the trials that have already been conducted? Certainly, we need to consider and really get good weight-based strategies, allometric scaling if that's necessary, but that's all going to come from PBPK and our good model development. You know, there should probably be more consistency in the endpoints that we choose, and if we're going to choose different endpoints, there should be a justification. I think the idea of an external control based on pooled placebo is a very interesting idea and could be used, particularly maybe even in new agents. We also heard that, you know, there are reluctance of families to enroll if the safety is not clear. All these things and long-term safety that we have to consider. And then really, if we're going to do a good job and lessons learned from previous trials, we have to do a better job of communicating what the goal of the trial is, making sure patients and families, the family unit, as I heard, have bought in, and that they have been at the table in the design of these trials. Next slide. So modeling and simulation, I think we heard a lot about why there is particular interest and importance in that less than 1 year, less than 2 year age, because of that developmental ontogeny of, metabolic enzymes. We also heard that we have great methods now that have been advanced over the last couple of years in terms of PBPK and POP-PK modeling that greatly have reduced, the idea that we do this intensive PK sampling studies that we used to do 10 or 15 years ago. Next slide. Newer trial designs, we heard a lot about that. I just listed them here, but I think the most important point is that last one, that whatever novel or not novel trial design you want to use, really it should be centered around the patient and the family, and getting input on them. And throughout the course of the design of the trial will only, I think, help in increasing enrollment, retention, and buy-in after the drug is approved. Next slide. Okay, last couple of slides. These were the gaps in knowledge that we identified in this past panel. Look, we have confidence in the ARBs. We have confidence in, we have confidence in the ARBs, but we have gaps in knowledge about exposure responses for other drug classes. We don't have comparative efficacy. We don't necessarily know safety in terms of specific concerns of special interest like neurocognitive effects and long-term safety. And we don't know a lot about combination therapies or versus maximizing single agents. How does dosing change as a patient grow? Is it strictly based on allometric scaling and weight, or are there other factors at play? But despite those gaps in knowledge, I think the clinical trials conducted to date demonstrate that the drugs that work in adults also worked in controlled pediatric clinical trials. So what types of studies can be designed to address these gaps? We heard about PK studies in prevalent population, 1 to 6 years of age. That we can include, we should be including patients down to 6 years of age in all new antihypertensive drug trials. We should be using large databases to collect and coordinated registries to collect safety data like PedsNet. Importantly, I think the idea came up in new trials or new agents. We might consider how we might collect, and Eliza gave us some caveats, but if we have all this pooled placebo-controlled data, can we use that the placebo arms pooled from those previous trials? If we know, and if we think the treatment effects are similar, it might be a reasonable consideration. And then, one last, if we go back, so how do we evaluate specific certain risks? And then what about neonates? I didn't fill in anything because that's still the open question. Next slide. Last thing, what infrastructure, what operationally can we do to increase the efficiency of these trials? We talked about decentralization, doing some of these blood measurements at blood draws at home, involving families in the trial design, minimizing discomfort, and improving and maintaining good communication between patients and the clinicians, the nephrology community, and the clinical trialists. So last slide. Thank you for your attention. We will see you tomorrow. Thank you very much for staying late, and have a great evening.